Hey, hi everyone. Just a sound check. Everyone can hear me? Perfect. My name is Beatriz, and I am a bioinformatician at the Ontario Institute for Cancer Research, or OICR for short. And today I will talk about how our group maintains and updates our cancer genomics analysis pipeline while upholding our accreditation requirements. I will start with some background about the genomics program, and then I'll describe our clinically validated analysis pipeline and talk about our infrastructure. And the last section will be on how we maintain the pipeline and perform validations. So my group is part of the Joint Genomics program, which is a collaborative effort between OICR and the University Health Network, which has the goal of supporting clinical cancer genomics projects. And we offer CAP-ACD accredited ISO compliant services. In this slide, you can see some of my coworkers in the lab doing library prep and sequencing. And at the bottom right, you can see the building where we're located at the Mars Center in downtown Toronto. And we are one of the labs that offers these 3 accreditations, which allows us to support clinical trials. So a little bit about our accredited assays. We offer whole genome, whole transcriptome sequencing, and we also offer a few other assays like targeted sequencing, plasma whole genome. And if you scan this QR code, this will take us to our services page. But for the purpose of this talk, I will be focusing on the accredited whole genome and whole transcriptome assay, which I will refer to as WGTS assay. So this assay involves sequencing a combination of the tumor and normal whole genome and the tumor transcriptome. And this is followed by several QC steps as the data goes through our validated analysis pipeline. We then generate an integrated report, which is reviewed by trained clinical interpreters and a certified geneticist. And the clinical report uses data from this analysis pipeline, and it summarizes clinically relevant information about the patient's cancer genome. The goal is to utilize complete genomic and transcriptomic data for precision medicine, and so that effective treatment may be used in accordance with the genetic profile of each patient's cancer. So what does it mean to have a clinically validated analysis pipeline? Our group actually began as a bioinformatics core center, offering bespoke sequencing services and data analysis services. But we then embarked on this journey to become an accredited facility and providing our accredited assays. So this journey involved a validation of our whole assay, including our analysis pipeline. And the goal is to demonstrate the assay performance in terms of accreditation, in terms of accuracy, precision, and reportable range, and demonstrating a normal range. So in order to validate our analysis pipeline, our group performed an extensive validation project using 50 sets of samples with orthogonal data, which were sequenced and analyzed through our pipeline and evaluated in the 4 categories above. And this large validation effort involved selecting the right software tools that we were going to use, developing protocols, looking at the reports generated from our pipeline against the orthogonal data, and keeping thorough documentation throughout. As part of this validation, we are committed to do version control of our workflows, which are written in the WIDL language, and we maintain them in GitHub and version control. We also maintain pipeline version history and do code reviews and automated testing. So what does that pipeline look like? This is a diagram of the pipeline. The starting points are the whole genome and whole transcriptome sequence data, and the data goes through over a dozen workflows that are written in WIDL language, each of which uses multiple software tools, so I've called them mini pipelines, and they are version controlled in GitHub. Briefly, if you look on the left, the whole genome goes through alignment, and then a series of tools are run to estimate purity, ploidy, and copy number variations. We also call somatic variants and calculate tumor mutation burden. And then the whole transcriptome also goes through alignment and is used along the whole genome data to call fusion events. The whole transcriptome is also used to estimate gene expression and immune expression. So all of this knowledge goes into our open source tool developed in-house called Gerba to generate a clinical report. And I just wanted to say there will be a talk on Gerba, and if you want to know more about this tool, about the clinical reports, the talk is tomorrow at 11:20, so watch out for that. A little bit now about the analysis pipeline infrastructure. So this is a fully automated process from sequence to analysis, and we have monitoring systems in place for pipeline management and maintenance. And this is the first half of a diagram of our infrastructure, and it is a simplified diagram. So the first half here, we receive the samples on the bottom left, and they're worked on in the lab to extract DNA or RNA. And all the sample information is tracked in MISO, which is our LIMS system. The libraries then go into a flow cell, which is loaded into a sequencer, and the sequencers write directly into our storage platform or Isilon. And a separate system scans Isilon for new sequence data and updates the LIMS system. And we're going to follow the part here on the right on the next diagram. So we have MISO and Run Scanner, which talk to GESMU, which is an automation system for pipeline operations that was developed in-house. It scans the systems for a list of files that have been produced and uses decision action blocks to decide what actions should be run. So in this case, an action would be launching a workflow. And GESMU works with BiC, which is our workflow engine service that allows us to track output files and metadata about them. Then our next system is VIDAR, which schedules workflows using Cromwell, and it collects the output from these workflows and stores them in their final location and continues to track the files and their metadata. And Cromwell finally is our workflow engine to run analysis workflows from our pipeline. The point that I really want to make, I know I talked about a bunch of different systems, but the point is that data moves smoothly from sequencing to analysis without us having to do any manual work other than the data entry. So the data writes out from the sequencer, where it's regularly scanned and immediately picked up by different systems, which make decisions about which pipeline and which workflow needs to be run. It executes the workflow, and it tracks the files and the metadata about them and the sample provenance. Another point I wanted to make is that for each one of those systems, we have a development instance, which are set there to plan and test new features. And we've leveraged this system to develop our pipeline and implement changes and run validations. So this is the environment where we do our validations, and it's identical to our production environment other than the feature we are testing. Okay, so that was a little bit about the infrastructure, and now I want to talk about maintaining a validated analysis pipeline. And I'm going to start with monitoring that we do in-house. Okay, so in order to do monitoring, we've selected a few of the samples that we use for our initial validation. So I mentioned that we used 50 sets of samples for the initial validation. So we've selected 6 benchmark samples, which we repeatedly use to test our pipeline. So these cases were selected to represent different components of the clinical report. So they had interesting fusions, HRD or MSI, and actionable variants. And what we do is to analyze these 6 samples on a weekly basis. The goal of monitoring is to ensure that data analysis is consistent with our validated assay. And because we have a complex system, we want to make sure that nothing has been unintentionally altered that would affect the report. So one of the processes that we have in place is the weekly analysis of our benchmarking data. So if you recall, I mentioned we selected 6 benchmarking samples. And what we do is inject those into our WGTS pipeline, and we review the runtimes of the analysis pipeline. So the next graph here will show how we monitor this on a weekly basis. So on the x-axis of this plot, you have the date, so the week that we're looking at, and on the y-axis, you have turnaround time. And each of these box plots represents a step in our pipeline. So for example, the first box plot here in purple is demonstrating sequencing to alignment to QC turnaround time. And the last 4 boxes are different steps in our invariant calling. And what we're looking at is for our pipeline to be stable. So the first 4 weeks here, we see about the same time taken to run our pipeline, whereas on the next 2 weeks, we see an increase in the turnaround time. And usually we will ask the question, why is something going on in our pipeline? We usually understand what, in this case, there was maintenance done in the cluster, so that caused some delays in our analysis. But we like to keep a close eye on the pipeline. Okay, so we've injected our samples in our pipeline, and then we also run Jerba, if you recall, to generate clinical reports. So we generate 6 clinical reports, and then we compare that to a reference. So the idea is that we have a reference report, and we're looking if there are any unexpected differences between the two. And if there are any, then we investigate them. And we also have a set threshold in case there are minor differences that are not clinically significant. And how we do that, as part of Jerba, there is a compare script that allows us to compare 2 clinical reports in JSON format. And this will flag whether the reports are equivalent, identical, or not equivalent. So in this case, the reports were not equivalent. So if we look further down, the difference was an additional gene alteration that had an associated treatment for MTAP gene. And this is what it would look like in the report. So the top report was missing that MTAP treatment, and the bottom one does have it. So we would go in and look, why are we seeing this additional treatment option? So in this case, we looked at the database that we use to associate variants and treatment options, which is OncoKB. And there had been a new release of the database which had added this variant and associated treatment option. So that explains why we're now seeing it in the report. So it was a simple explanation in this case. Okay, so now I'll get into the last part, which is updates and validation, which is a lot of the work that we've been doing recently. So there are many reasons why we have to update our pipeline. And for each one of those updates, we have to decide whether we need to do a validation study to revalidate our pipeline. So here are some examples. The first example that I put here is pipeline optimization. So in this example here, if we update the memory allocation of a workflow, we don't require it to do any validation as it wouldn't affect the data. The second example that I added here is workflow optimization. So again, this would be a memory scaling task that was added to a workflow, for example. And we have implemented in our workflow development in Widow regression testing, and that allows us to test the workflow on a small data set. So if we don't see any change in the regression test, then we're confident that we don't need validation, as well as we know that the type of change would not affect the data. And the last 2 are examples that would require a validation study. So if we do an update to our pipeline, for example, updating the version for Delhi, usually that would mean the regression test would change. So we know for sure we want to validate this change. And the new workflow, introducing a new workflow would require a more in-depth study. For example, we recently introduced Purple to replace Sequenza. And this would definitely require validation. And I added a column here to also show that we need to update our documentation anytime that we update the pipeline. And the example that I'm going to talk about is when we updated the Delhi version. So I said that we've leveraged the system to now inject benchmarking samples in our production environment, run them to our pipeline, and generate clinical reports. What we do is we take our development instances that I mentioned, and we install the same pipeline and update our software there. So Delhi here has a star, so we've updated the Delhi version. And then we run Jerba and generate the clinical reports and compare the 2 versions, the one with the updated Delhi version and the old one. And we look for any differences. So in general, we are looking for, generally we don't expect any major differences, but we look for consistency of results in general. And if we are happy with what we see in the reports, then we move this new version into our production environment and we update all of our documentation. And now we release our new pipeline version. So really, that's the whole story that I wanted to talk about here today. And just very briefly, I just wanted to say some of our future work. So we are looking at Dragon as a way to speed up our pipeline. We're also working to 2-week turnaround time, WGTS. Currently, we have a 45-day turnaround time from sample receipt to the clinical report. So we're looking to reduce that drastically to 2 weeks. So that is just very early, but we're looking at it, as well as introducing HLA typing into the clinical report. This is our GitHub page where you can find all of our workflows. And with that, I just wanted to thank everyone that has worked really hard on this infrastructure and the pipeline and the work that I presented here. And again, there will be a talk on Jerba. There are also 2 other talks if you're interested in how we are dealing with the amount of data we're producing. So last year we produced 1.4 petabytes of data. So there will be a talk on that, as well as a dashboard that we use to track our samples. So those are today in the Bioinfo core. So thank you.
Thank you, Beatriz. Does anyone have any questions? You can hop on up or type it in the chat if you'd rather.
Hi, great talk. So actually in relationship to this morning's keynote and to actually Morgan's question, are you going to move to a pan-genome reference as opposed to doing, I'm assuming you're using HG38 right now?
Yeah, we're using HG38. That's a great question. We've talked about it, and I think we were all really excited at seeing that we could still have a linear genome and but take advantage of some of the pan-genome. So I'm sure we will look at that after the keynote.
A remake of all the pipelines, I guess.
Yes.
It's a
so
big task.
It will not be a quick adoption that will happen, but we're definitely thinking about it.
Okay, thanks for sharing your experiences here. I was just wondering whether, how frequent do you make a new release, and do you have concept of like major release, minor updates, etc.?
Yeah, thank you for your question. I would say we update our pipeline with major changes, maybe about 4 times a year, just the number there. But we do have a concept of minor versions. So if we know that the change that we made to the pipeline will not affect the data, we just keep a record of all of the software version. And we'd record that in a single document. But if it's a major change, then we actually run the validation study and we update the pipeline version. So I hope that helps.
Okay. Yeah, thank you.
We have 1 minute for a very quick question.
All right, a quick question. So it's about the benchmark samples, the 6 that you said. So I'm not sure, maybe I missed this. Are these synthetic data sets or they're actual real data?
No, it's actual real data where we have orthogonal data. It was used initially in the validation study with the 50 samples. And then from those, we chose 6 of them. Yeah.
And was there any like specific requirements or something to make sure that these 6 can be used, like, I don't know, as long as you want, and, you know, maybe, I guess, like use on developers' machine who work on these pipelines and do those things? What makes it that it's okay for these 6 to be used like for the whole development environment?
That's a really great question, and I think I'm gonna reach out to my team and maybe talk to you afterwards, because I think some of my teammates might be more familiar with that decision.
Thank you.
Thank you.